Hi, my name is Adair Osterly, and we're going to discuss the beveler and how to bevel a pipette, and firstly, how to actually put the beveler together. As Jan had mentioned, the beveler is the first product that Sutter made, and the story goes that as long as we sold 12 bevelers a year, we would keep the business running. So that was back in 1974. Obviously, we continued to sell 12 bevelers a year, and we still do now. So we're still up and running, but we actually have a lot more products in our catalog. So let's proceed here. We are going to be making some sharp and hopefully beautiful pipettes. There are ways to make really ugly, terrible, awful ones. So we'll go over that too. Not that that's your goal, but it is a goal to avoid that. So the outline of what I'll be discussing, I hope I'll hit all the topics. I made a lot of slides, and we'll see how fast I can go through them without having you guys try to run behind me to keep up. So first we're going to go over what is beveling and the applications that require beveled pipettes, assembling the beveler, how to find the frigging pipette. It is very hard sometimes to find the pipette. And I have a few tricks that'll help you find it and make the process hopefully enjoyable rather than a pain. Lighting, the lighting is very important. The earlier bevelers, the first bevelers we sold, did not have their own light source. We did eventually add a light source, but it was a bit weak. And now with LEDs, we have a very bright light, and that helps. You can also add a light yourself if that is required, but we'll go over that. We're going to go over the glass, the tools, and the solutions. Beveling small and large tips. How long does it take to bevel a pipette? Beveling at various angles, and then discussing the taper length of the pipette that you can actually bevel. If it's too short or too long, you're going to run into some issues. So you're going to go from ideal to impossible. And then discuss the tip sizes, what's doable and what is challenging. What is beveling? Beveling is when you're grinding the glass at a diagonal to create a sharp and angled and more durable micropipette. Here's an example of a beveled pipette. Isn't that gorgeous? And then you'll be using the Sutter BV10 beveler, which is shown here. And you have a light, and there's a microscope. The beveler doesn't have to come with a microscope, but it's very hard to use it without it. And you really need it at that angle, too. Dissection scopes, you might have something on a post that swings over. People have tried that. It's not very useful. So it's really best to get the scope with this beveler. And then they also come with a meter if you're going to be doing electrophysiology. I will not be discussing meters and the use of the beveler for electrophysiology, but you can call Sutter if you need more information on that. Here is the preview here of beveling a pipette. I'm going to show this. It's a fast motion, so you can see it. It's not always as fast. It usually takes many seconds to maybe a few minutes. But isn't that gorgeous? The next thing we're going to go over are the applications that require beveled pipettes. Beveled pipettes are required when the tip must cut through thick tissue, through a tough cell wall, or an egg that has a fibrous membrane or a chorion. And examples of these applications that it's required for would be brain and spinal cord injections, Drosophila, mosquito eggs, shrimp eggs, fish eggs like Medaka, moth and butterfly eggs, plant cells, wood cells, and pollen, zebrafish, and frog eggs. Don't always need them for that, but usually people just trim the glass back and they don't need to bevel. But if you want to bevel, you can, and the pipette lasts longer. Adult insects, they can be used for cephalopods, the eggs of cephalopods, octopus, squid, and cuttlefish. Pronuclear and cytoplasmic injection applications like CRISPR, when you're going into a mouse egg, or you're doing nuclear transfer or stem cell transfer or ICSI. The glass types that you can use are borosilicate, aluminosilicate, and quartz. Borosilicate is a good glass, a little softer glass than some of the other glasses I'm going to show here. Aluminosilicate is better because it's 10 times harder than borosilicate. And then quartz glass is best, and it's much harder than borosilicate, 15 to 20 times harder. It's the most durable glass that is available. It's easier to bevel because it's so stiff and it grinds back really quickly. It stays sharp longer and it won't dull as fast. When I was doing some microinjection work with mosquito eggs, I think we hit a milestone of 537 injections with one pipette. And then it finally broke or got dull and broke. So it was a sad moment, but I was very thrilled that it lasted so long. And then everyone who's going to be using quartz glass must use a P2000 laser puller. And that has enough heat to melt the quartz. The P97 and the P1000 pullers don't create enough heat to pull quartz, so you'll need a P2000 puller. And these are some glasses that I recommend. There are lots of different glasses we offer: thick wall glass, thin wall glass, filamented and non-filamented. And then here are the examples of the pipettes, what they look like when they're beveled, and the glass that I'm using for this. The top is thin wall, then you have thick wall, then there's solid rod glass, and then there's filamented glass, which is required when the tip is small. The filament in the glass helps create capillary action, so when you backfill the pipette, the solution goes to the tip without forming bubbles or not flowing to the tip at all. Okay, we're going to get started here with assembling the BB10 beveler. Here's the beveler base, and there it comes with a base and power cord. And there's the newest version of the light that we have. And it's focusable, which is kind of nice. And that is the top of the pedestal pair. That is the part that holds on to the beveling plate. And then here are all the parts that come in the box. Again, the beveler and the power cord right there. You have the beveling plate holder. You have the micromanipulator right there. There's a wick holder and a wick right there. There are two beveling plates that come with the beveler, and you can select from 4 different grit sizes of beveling plates that I'll review soon. We also have the microscope and the mounting rod and hardware. And you can also optionally get 20x eyepieces, which allows you to see at 80 times magnification, which is best for tips that are under 10 microns. Those are the eyepieces there. And then last but not least, the degreaser and the beveler oil, which you need for cleaning, and also the oil is used, as you'll see soon. Additional supplies you might need are here. As you see, I've labeled the far left bottle of ethanol 76.875%. That's so no one takes it from me. It's different, and they don't think it's something that they should use, so they don't take it from me. But anyhow, there's the other supplies that are there. And I use medical grade water and medical grade ethanol. They don't have any additives in them. And for cleaning of the glass, you would use ethanol and water. And then for the cleaning of the beveler itself, the parts, you're going to be using water and maybe a degreaser and ethanol for the parts that are oily. And then the top beveling plate, you're just going to rinse it with clean water. So assembling the beveler, the first part is putting on the eyepieces. It comes with 10x eyepieces. If you bought the 20x, you can put those on there. The little Phillips head screwdriver you would use. And here are the mounting rods and the mounting block and the mounting post. And first you put the mounting post on and you put it on super tight because you're going to be using that as a pivot point, and it's going to hold on to the microscope. This is the mounting block. The line right there goes up and towards you. And so that needs to have the proper orientation, and I'll show you soon why that is the way it needs to be installed. That's the microscope mounting rods, and then you're going to assemble that, and you're going to put that together super tight. Step 2, you're going to slide the rod into the mounting block, and you're going to line up the line there you see with those little divots. And that will create a situation where the microscope is at the right angle for the best viewing of the beveling plate and the pipettes. And so you're going to tighten that super tight, and you're going to tighten this tight. It is a pivot point, so you want it to slide a little bit there, but not have it too loose. You want some control. Step 3, you're going to remove the cap. You're going to loosen the knob, and then you're going to slide the post through the hole, and you're going to mount the scope, and you're going to slide the scope all the way down to that ring stop shown here. You're going to tighten the knob to lock the microscope in place, and then you're going to adjust the focus knob on the scope so it's flush with the base, and that will be a good starting point for you right there. And then you can put the cap back on, and then you're going to put on the wick and the wick holder. Step 4, you're going to select a beveling plate. We have 4 different grit size beveling plates, and they go from coarse to extra fine, and they all are color coded: copper, lavender, gray, and sage. Copper and lavender are the ones I use most often, and you would select the beveling plate based on what tip size you're going to be beveling to. You're going to remove the retaining ring, and then you're going to put the beveling plate onto the bottom part of that assembly, and then you're going to attach the retaining ring. And you want to tighten all the screws snug tight. And I usually go around like I'm tightening the lug nuts on a tire. If you've ever changed a tire, you're going to go around and just do a little extra tightening all the way on all 3 screws so it's evenly tightened. Now, step 5, we're going to continue with the assembly of the beveler, and we're going to add some oil to the top of the optical flat. And the beveling plate is floating on that optical flat on oil. And that's what makes this beveler so amazing, is that it's rotating, and it's rotating perfectly flat, and there's zero up and down vibration or movement of the beveling plate. So you only want to put 1 to 2 drops of oil. If you put more drops, you're going to have an oil slick. So don't do that. You're going to have a crazy oil spill. And you're going to turn the power on first, and then you're going to actually put the beveling plate on top. And I'll show that to you. If you assemble the beveling plate on top of the bottom base, all the oil will go away. And if you put the beveling plate on top and you don't have the beveler rotating, all the oil will go away. And when that happens, the beveler starts jerking. So you want to avoid that. Step 6, we're going to get the manipulator out and set up. You're going to remove the strap. This is what holds onto the pipette. You're going to make sure that the toggle plate is in the proper position, like that. That is correct. That is wrong, and that is wrong. The plate has flipped over. So you want to make sure it's in the correct position. And then you're going to adjust the pivot point to position number 1. That's a very good position for when you're adjusting the fine dial for lowering the pipette. It's a nice gentle rate of lowering of the pipette onto the plate, the beveling plate. You're going to dial the coarse knob all the way up, and you can see that the angle plate holder is almost flush with that top part of that assembly. And then you're going to dial the fine focus to around 9.5. That's where that toggle plate right there is level, and that's what you want. You want to make sure that you start with that every time you're starting to bevel a pipette. You want to re-level that with the fine dial. The next step here is creating the angle that you want. So you're going to adjust the angle plate, and then you're going to lock it in place with the locking screw. And then you're going to put the wick off to the side on the beveling plate. And then you're going to add water, which has a little bit of detergent or photo flow in it. Why do we do that? Because that breaks up the surface tension of the water. And so the water will lay flat on the beveling plate, and it won't puddle. If it's puddling, you're beveling dry and wet, and then you'll end up with a lot of dry glass particles in your pipette, which will clog the tip. So you always bevel with a wet surface, and that is what the wick there is for, and that is what the water is for. The water you're going to add, so it's about 1 millimeter thick. You're then going to position the manipulator onto the beveler base plate, and I usually mark it with either a Sharpie or some tape. So that is the position I like to have everything stay, so I can find my pipette more easily when I go from one pipette to the next. We're going to also go over here quickly the taper length, tip size, and the time it takes to bevel a pipette. 3 to 5 millimeters is a bit too short, usually because you have a high cone angle. It's like more like a patch pipette, and the tip will get beveled too fast usually, and it'll be hard to bevel to the tip size you're really wanting. 5 to 10 millimeter taper length is ideal. 10 to 15 can become challenging because the glass becomes a little too flexible. And then over 50 millimeters, it's possibly impossible because it's way too flexible. So here's some taper lengths here. The first one being a little too short. The next 3 in a row are doable, and then the one at the end, that would be pretty much impossible. But those taper lengths that are really long are the ones that you're normally going to be breaking back to make, say, a 15, 25, 50, or 75 micron tip. So you pull along, and then you'll be breaking them back. The tip sizes that are possible to bevel are under 0.5 microns, but it's hard to see. 1 to 25 microns is easy, no problem. 25 to 75 microns is easy, no problem. And then when you get to the tips that are larger, it's doable, but it takes longer. And then once the tip is really large, you're going to run into problems because it's too big and there's too much glass volume. the time it takes to bevel the pipettes, anywhere between 5 to 60 seconds, depending on the tip size that you're aiming for, and then up to 1 to 5 minutes if the tips are really large. If you go to the webinar titled Achieving the Impossible, 17 minutes and 55 seconds into that webinar, you will see information about how to score and break your glass to get a clean break before you bevel large tips. You need to get rid of all that glass first, that's smaller, and then you will put the tip on the beveling plate to bevel. If you're making a small bevel tip, you just pull it and you just bevel it. You don't need to cut it or trim it or forge it. But if the tip is somewhere in between sort of a small and a large tip, you do need to forge it, the 5 to 15 micron range. You need to cut it or trim it with some tweezers or scissors, or you need to use a microforge, and that cuts the glass at the tip size you need, and then you would then bevel it. Again, that's usually between the 5 and 15 micron tip sizes. Okay, now we're going to bevel the pipette. Step 1, you're going to put the glass into the holder. There's a little clamp there, rubber washer, and then there's little grooves that you put the glass in. I always place the glass at the same location on my index finger, middle of my pad. So I'm always loading the glass at the proper length. So when I drop the glass down and I look through the microscope, I can more easily find the pipette tip. So right there, you see the glasses against the pad of my finger. Then I always have the finger touching the black block before I tighten the clamp. Then I'm going to use the coarse dial to lower the glass to the plate to be just above the water or barely touching the top surface of that water. And there's the coarse dial right there that you're going to use. So here we are, and we're turning that dial clockwise to lower the glass, and then you'll see where when it finally touches the water, you'll see the water actually go into the pipette there a little bit too. The next step, what you want to do here is you want to find the pipette. That's why I titled this How to Find the Friggin' Pipette. It's very hard if you don't go through specific steps. The number one trick, the first thing to do is to focus on the back edge of the retaining ring, right there where the blue arrow is. You're going to focus on that. You're going to push the scope forward until you see the black edge, and you're going to focus on that first. This is like focusing on the edge of a water drop to find the bottom of the water and where the surface of your slide or the Petri plate is. And now I'm going to slowly pull the scope back towards me and maybe slightly pivot it right to left to find the tip. So I'll show you that right here. I'm just pulling back a little bit, pushing forward. This is how you're going to search for the tip. You're going to also maybe toggle the tip, pivot it actually right to left a little bit to find it. This is what you do not want to do. Do not tilt the scope forward or tip it back. If you do this, the pipette will get lost. You'll get lost, and you might just go crazy trying to find the pipette. So don't do that. The next thing you're going to do is when you've found that edge of the retaining ring on the beveler, you're going to slowly pull the scope back towards you to find the pipette, the reflection, or the shadow. You'll see something eventually, and you'll focus on the pipette. Don't let the reflection or the shadow fool you, and I'll show you what I'm describing there. Then you're going to adjust the light. You're going to slightly pivot the scope right to left to get it centered in your view, and then you'll refocus, and then you'll lower the pipette to the surface. And I'll show you that right here. I'm pulling the scope towards me. I see a little bit of a shadow. I see a reflection. It looks like the pipette's aiming up, but that's actually the reflection of the pipette. So I want to focus and find that pipette that's above. There it is. And now my reflection at the bottom is now a shadow. And now I'm going to lower the pipette so it's just touching the surface. And the lighting is key here, and it's sometimes a challenge to find the right lighting, but I'm going to show you the bad and the good lighting. And once the lighting is adjusted and you have the proper amount of water to get the meniscus out of the way, and you can see through the water, you'll be able to see the pipette. And you're going to use the coarse dial to touch down to the plate, just barely. And then you'll use the fine dial to gently touch the plate and then advance the tip downward so you can bevel the pipette. And you're going to look for a very slight bend or bow of the pipette, very slight. A quarter turn usually gives you 0.5 to 3 microns if your pipette has not been trimmed or cut back. Half a turn gives you 3 to 5 micron tip. 3/4 of a turn is good for a 7 to 10 micron tip, but sometimes you might need to trim it first. And then for tips that are over 20 microns or larger, you're going to need to, again, trim it back, and then you're going to do usually 1 full turn, maybe 2 or 3 if the tip is really large. This is the fine dial, and you can see I've marked it, and I've put the tip sizes there just as a reference. But usually I have that front mark there where it says 20 microns. I have that front mark there all the time, so I know how many parts of a rotation or full rotation I've taken. And then here is a video showing the pipette. I'm moving the lighting around. I can't really see much at all, and I'm just moving that lighting. Oh, that's good. And I'm just going to move the lighting until I get something good. Once I touch the water, the meniscus is in the way, and then I can't see anything. So that's going to drive you crazy. So then you need to move the lighting around a bit more to see through the water. If you can't see through the water, you're going to add some more water so that meniscus raises higher and is out of the way. So you'll see that eventually. I'm still playing with the lighting here. You'll see how hard it is. Okay, now I have a good lighting, and now I'm going to add some more water. And that'll bring that reflection, that meniscus, above the pipette. I'm going to add a little bit more. Now, my water in this situation wasn't totally clean, so you'll see a few pieces of crap fly by, but you'll see what that's like. And then when you touch the plate, you'll see the meniscus of the water inside the pipette go up the pipette. See that happening right there? Isn't that cool? That's when you know you've touched the bottom of the plate. And now you see a little bow there. That's when I just put like a quarter turn or a half turn on that fine dial to push onto the plate. See, there's a little bit of dust that was in there. And then after about 15, 30 seconds of beveling, if I'm beveling a small tip, I'm going to raise it up with the fine dial. And then I'm going to raise it up further with the coarse dial to remove it. There you go. Now, if you push too much, here we are beveling some large tips, but I'm showing you the proper amount of pressure and then not the proper amount of pressure. So 2 to 3 full turns is good on the fine dial if you're beveling a really large tip. 4 to 5 rotations is too much, and it's going to be somewhat disastrous. So try not to do that. When you push too hard, it changes the bevel angle. You end up with a tip that the absolute end of the tip never sees the plate, and you get the squared off blunt tip. I'll show that to you here. And the tip will fracture, it'll chip, it'll break back. You might start crying. So don't do that. So here we are, I just touch the plate. I'm going to drop it down. Now I'm going to use a fine dial, and I'm going to make 1, 2, and then 3 full rotations. Now it's beveling. Now, the trick is here to be patient and not sit there and try to push more on the plate because you're tired of waiting. It might take a minute or two here. And once it actually has completed the bevel, it's good to lift it up gently with the fine dial right when it's done beveling. Here I'm showing you pushing even further. I went 2 more rotations down. Now you can see how I've changed the bevel angle, and you see how the tip's not even seeing the plate. The back part of the glass is beveling, and the tip is not seeing the plate. It's just staying as this little blunt, broken, or actually not really broken, but just this blunt tip. Now it starts to break, and there's just so much pressure, it's chipping. You might think that thin part in the front is really nice and sharp, but it's so thin, it's fragile, so it'll break. So beveling at a really low angles, like 15 degrees, I really advise people not to do that. And now I'm raising the tip back up to get something more reasonable. And then once I've got a nice clean bevel there, I'm going to stop. The other thing might happen when you push too far down on the plate. Remember how the absolute tip wasn't seeing the surface? Well, that happens on smaller tips too. And when that happens, you get this little spike at the end of the pipette. Now, that spike is really just a flimsy spike, and the tip is pretty blunt once it breaks off. So you don't want that spike. And when you bevel aluminum silicate glass and quartz glass, you're more likely to get that spike, especially if you push down on the plate too hard and too quickly and too far, or you're beveling at too low of an angle. So you really want to avoid that. Step 5, Here's what I do to remove the pipette and install a new one. I don't want to move the scope. I don't want to move the manipulator. I don't want to move the light because I spent so much time getting it perfect. So I'm going to reach in from the side and I'm going to spin that little locking screw, and then I'm going to take the pipette out. So again, don't move anything. Gently reach in from the right side. Again, not disturbing anything. And then spin that little locking screw and then take the pipette out. And then put a new one in, and you'll see I've got the pipette at the same location in my index finger. I put the finger up against the block, and I've tightened it. Then it's at the same location. I'm at about 12 o'clock, and it's not advanced too far forward or too far back, and I can drop it straight down and find it. Now, once you've made your beautiful pipette, you need to clean it because it's got all the solution in it and some glass particles. I use water, 75% ethanol, and 100% ethanol, and I clean it through a series of solutions. I'll aspirate and then eject the solution, maybe about 2 or 3 times in each solution, the water, the 75% ethanol, and then 100% ethanol. And then that's my setup right there. I'm using the Sutter XenoWorks digital injector, and I'm using that for nice control to make the pipettes. The other thing is, this is really nice. If you have an injector, you can use the high pressure to blow out the solution, but you do need to be a little careful, and I'll show that to you in a second. You're going to need to clean up afterwards. Yes, you need to take the entire system, not the entire system apart, but you need to take the top pedestal piece off. You need to remove the retaining ring and the beveling plate, and you need to clean the bottom of the beveling plate with 75% ethanol or degreaser. Do not get that oil or the 75% ethanol or the degreaser on the top of the plate because it might ruin the surface or wash off the diamond granules. So I'm washing off the bottom of the beveling plate, and then you're going to clean the top of the plate with pure water, shown here. And then you're cleaning the optical flat too. Don't forget that, clean that off also. And that would be with ethanol and or degreaser. So you want to put the beveler to bed after you're done, which means after you clean the plate holder and you clean the retaining ring, you're then going to lay out the parts to dry on a bed of paper towels, including what I call the virtual screwdriver, since we're talking about virtual reality here. I did that because I forgot to put the screwdriver there. And it's good to keep one there at all times so you're ready to put everything together and bevel. So keep your virtual or your real screwdriver off to the right side of your parts. And then cover the parts with a Kimwipe blankie because, you know, we're putting it to bed. And that keeps everything dust-free. And then you're going to do the same thing. You're going to cover the optical flat with a Kimwipe. And then you're going to return tomorrow for more fun beveling. And I'm going to just show you some hazards here. This is kind of fun, but I have never actually injected anybody accidentally or driven the pipette into anybody but myself. And the pipette sometimes does go flying if you're using high pressure injector to clean it. So you need to point it away from you. And one of the hazards is stabbing yourself. And the other hazard is flying pipettes when you're cleaning it. So I just want you guys to be very careful if you're using a high pressure injector to clean your pipettes. Here is an example of me having accidentally stabbed myself with a beveled pipette. It's gone pretty far in. It did come out whole though, so I was lucky, but I did draw a little blood. Hopefully no one's too squeamish to see that, but it's a good story to tell. And here's the beveler in slow motion, high pressure. You will see on the left, bottom left, there it goes. It went flying because I didn't tighten the chuck tight enough to keep the tip from sliding out. So you really have to tighten those a lot. The other example here is a drawing of my best friend, and nope, see, I accidentally stabbed him in the eye. So you do want to avoid that too. Anyhow, I had to have a little bit of fun at the end of putting these slides together. And again, if you don't have all this equipment and you don't have the time or the expertise and you want to just buy your pipettes, you can do that too from Sutter. We do make a variety of custom pipettes.